just to explain the photo, so Oregon's different than Texas because we have a lot of mountains. In fact, our medical school is on top of a hill that's over the city, but we built up everything on the hill. And so the only way we could expand our cancer center was to go down to the river, but that would involve crossing an interstate freeway and multiple highways and going through neighborhoods. So our engineering solution was to build a tram. So that's what's shown here. And so the tall buildings down right by the river, those are the outpatient cancer clinics, and this is how I commute from my office, which is on the hill, down to the clinic. So this is actually interesting giving this talk after a break, because historically what I do in this type of talk is I talk about research we're doing in the lab, and I talk about 1 or 2 clinical studies that are coming from the lab or coming. But actually there are more open clinical studies for advanced GIST than at any time in history. I'm going to kind of walk you through that. Of course, since KIT is the dominant driver of many GIST, not all GIST, getting new drugs after imatinib, sunitinib, regorafenib, ripretinib is still a strategy. Build a better mousetrap. We can still do better. So we're getting more potent and selective tyrosine kinase inhibitors, which is the general type of drug that imatinib falls into. So we know that these tumors remain dependent on KIT. So if we can do a better job against KIT and fix that stuck gas pedal, then we can still make progress. But we also are realizing that there are other types of mousetraps that we could build, and that there's other strategies. And so really, for the first time in most of the last several decades, a lot of new, completely different treatment approaches are entering into the clinic. Historically, it's always been, we have drug 1, let's build drug 2. And then we have 2, let's do 3. And then we have 4, let's do 5. But now, instead of trying to be 5 or 6, people are saying, if we have a better drug, let's be 1 or 2. So there's significant interest in moving new drugs to earlier. In fact, there are currently 3 ongoing phase 3 studies to be the second drug, which is to replace sunitinib. And there's also, unlike previous conferences, there are a lot of new approaches to treating sdh-deficient GIST, and Dr. Siklik is going to talk about that this afternoon. So these are the new treatments that I will be talking about. Again, historically, if I showed this slide, it'd be like 1 or 2 drugs here. So I'm going to talk about second-line therapy because there's a lot of activity going on, and there's actually going to be or ongoing multiple phase 3 studies. So there's a study known as PEAK, which is combining a new drug, bezuclastinib, with sunitinib. There's the IDX42 drug, and there's ripretinib as a second-line agent. And we'll talk about that. And then later line therapy, there's also these studies which are ongoing. And as Dr. Samaya said, these are generally phase 1 studies where we're either trying to figure out the dose of a drug, or we're trying to figure out how to combine an existing drug with something new, because we don't know what the toxicity of either would look when we add them together. So I'll talk about these 5 drugs as well. So let's start first for second-line therapy. So to think about this, though, we really need to understand, if we have a KIT mutant GIST and we expect it to respond to imatinib, what happens during the treatment that makes it not work? So, KIT is not evil. There's a normal role for KIT, and so it's just supposed to be like a door. It has a function, and we would like to go outside through this door into the beautiful outside world. But when we get mutations that cause the GIST, now we have a problem. The door is locked. We can't go outside and enjoy our, the beauty of outside nature. And so how do we defeat locks? We need a key. And so we can use a drug like imatinib, and that can open the door, and we can go outside. Or in the case of Dr. Samaya, which I always use the exact same analogy to teach medical students, if the gas pedal is stuck, we need to, figure out a way to unstick it. And so that's great. So that works until something else happens. And then over time, due to random mutation, tumors can get a second or third mutation in KIT. And that's like, now we have a double lock. So we have a key that works on the first lock, but it can't unlock the second lock. So we still can't go outside the way we would like, but we can develop a new key such as sutent or sunitinib, but then they can be a different type of lock and we can use regorafenib. But over time, we run into more locks that we can handle, or the other way I think about it is like car keys. So you have a fleet of cars and they all had the same ignition, and so the cars are all running, but you could go out and turn off every single car and things would be fine, until you go out one day and you realize that half the cars, you can't turn them off. And that's what happens sometimes in patients with GIST that are resistant to imatinib. Some mutations that were controlled start growing and we need another key. And if one key could fix all those cars, would be great, but really what happens is you have 30 cars and you need a bunch of different keys and you only have 2, and so you succeed in some tumors and not in others. And so when you see your oncologist and you're on a drug like that and you have a scan or you read the report, you'll see, well, these 5 tumors are stable or smaller, but these 5 tumors got larger. And why is it? It's because it's different locks and because the drug is not the right key. So, and then I think Nita showed a version of this, that these secondary mutations, so the top red star, I guess for Texas maybe, is the original KIT exon 11 mutation, but then you can get a second mutation in where the drug binds, which is called the ATP binding pocket, or AP, but there's a whole nother class of mutations that can be in a different region called the activation loop, or AL. And the problem we have is that if you have this mutation combination, sunitinib will work great. If you have this mutation combination, sunitinib doesn't work at all. But there's a new drug called bezuclastinib, which is the exact opposite. It works on these, but not those. And ripretinib, or Kinlock, which is our 4th drug, works well against this, but not against that. And so really, these are the locks or the cars that we're trying to control, and our drugs are imperfect. They don't work against all of the mutations. And so our solution is either develop a new drug that takes care of everything, or take 2 things and add them together so that we can handle both. And so the bezuclastinib strategy in combination with sunitinib is exactly that. Sunitinib works against the AP mutations, but not AL. Bezuclastinib works against AL, but not AP. But together, theoretically, we're targeting all the mutations. So has been running now for a couple of years a phase 3 study in which patients get the standard treatment of sunitinib, 37.5 milligrams, versus a combination of bezuclastinib plus sunitinib. And originally 388, I think it's slightly closer to 400 patients. The study has completely filled up. All of the patients needed for the study have entered the study, but it's a blinded study. So the radiologists who are looking at the scans to decide if treatments are working or not, they don't know what the treatments are. Nobody on that side knows what the treatments are. And so until radiologists say that 2/3 of the patients or so have had a progression event, we don't unblind the data, so nobody knows the results. When will we get the results? I'll get that in a minute. This is just kind of showing some data. The drug had to move from one company to another, and then you had to remanufacture, and then you had to kind of start over and figure out the dose because the new tablets were absorbed much better than the old tablets. This is what's called swim lane plot. So each vertical or horizontal column is a row is an individual patient. If you have a green arrow on the far right, that means that the last time we have data on the patient, they're still going. Times on the bottom axis. And overall, the progression-free survival median was 10 months, which is pretty respectable because second-line median progression-free survival historically is 6 months. So this is quite a bit longer. Again, this is not the randomized part. This was, let's figure out if the new drug dosing is correct. If you looked at patients in the second line, again, this is a very small number of patients because this was a sort of the restart drug development. The median PFS was 19 months in only 6 patients, which is a really small sample size. But again, our expectation would have been 6 months. So that looks pretty promising. So where we stand right now is that the enrollment's completed, but we don't know any results until enough events happen. And so when will that happen? It wouldn't surprise me if it was 3 minutes from now, or 3 weeks from now, or 3 months from now. I predict that maybe around ASCO or sometime this summer we'll get the results. But until it happens, no one knows the results. It is widely predicted that this combination is going to be superior. I mean, you're comparing A versus A plus B. It can't be worse than A, right? Because it already contains A. So we think it will be better. So if it is better in a statistically significant way that the FDA would think this is a clinically and humanly important advance, it will become approved, which means in the future, if you have progression on imatinib, your choices would be to go on sunitinib alone or the combination. But you would imagine that everybody would take the combination because it would have better results, and you could always just drop the new drug if you thought that was the problem. We don't really know a lot about the combination toxicity. We'll know a lot after the randomized data has been reported. To date, if you showed me a table of people, side effects and frequency and severity taking the combination, but then cross it out and say this is sunitinib alone, I would believe you. So it doesn't look dramatically different, but that's just a gestalt. It's not truth. You'd expect there has to be a little bit more side effects, but having participated in the study, I would say that to me, it's not apparent that people have more side effects or worse side effects on the combination than is the general experience of sunitinib, which is Dr. Samaya said, some people are in 50 and some people are in 12.5, so there's a big range of tolerance. Another drug that's being developed and ultimately getting to the second line is IDRX-42. So this is earlier in drug development, so it doesn't have a name yet. Getting a name is very expensive, so companies don't spend money to get a name until they're pretty sure that they're going to go somewhere with the drug. So probably this year a name will be announced, but I don't know what it is. This solves the problem of these resistance mutations because it does a better job than any other drug that we've seen in a test tube. It's not perfect, but it's better than anything that we've seen. And if you do preclinically, like in mouse models, it looks better. And an important differentiator of this drug is that sunitinib, regorafenib, ripretinib have these side effects that Dr. Samaya talked about: hands and feet on fire, mouth sores, high blood pressure. All of that comes from antagonizing something called VEGF-R, which is really not important to the GIST response, or not very important, we think. And so by not having that side effect, that immediately makes this drug way more tolerable than even the standard agents, and kind of not necessarily, I wouldn't make a direct comparison, but it puts it more in the imatinib kind of camp of side effects versus what we see with regorafenib, for example. And anybody who's been on it, raise your hand. And anybody on it who had a lot of side effects, raise your hand. Should be the same number of people, almost, yeah. Right, so it's our least favorite drug because it has the most side effects, severity, and commonality. And in fact, the way we give the drug is designed like you cannot take it for more than 3 weeks because no human can take it for more than 3 weeks because the drug levels rise over time. They get really high. People get side effects, so you need engineered breaks. So IDRX, we think, would be a kind of in the idea of what we want for a drug: less side effects, more activity. And so this is some of the early data. So these are what's known as waterfall plots. So oncologists love this because it gives us a visual representation of everybody. So every patient in the study is their own little bar. And what we're looking at is a change in tumor size, and it's the best change in tumor size. So this person over here, the best result they ever had was the first time we did a scan, unfortunately their tumors were 40% bigger. So anything going up means that you never responded to the drug, not even stable disease, because if your best response was stable, you would be here. And every bar that goes down is response, but response is cumulative. So if your first scan you're down 10, and your next scan you're down 20, and your next scan you're down 50, what this bar will show is 50, because that's your best individual response. And then we have different mathematical ways, but in what we call RECIST, which is our standardized way of doing research scans, if your tumors shrink by 30%, which is the sum of your diameters, then that's extra gold star or extra black triangle, highlighting those patients. And this was a phase 1 study, so you can either think about all the doses, but we tested lots of doses from as low as 120 milligrams to as high as 1,200 milligrams, and we came back to 300 milligrams as being the dose that gave us our best balance of side effects and activity. And if you look at the objective response rate in the second line is 46%. By historical comparisons, sunitinib would be 9%, although in more recent studies, maybe it's more like 20% because we changed our methodology. But nonetheless, 46% is quite impressive. And so this study kind of has run its course in that we figured out the dose. And so the next step is to do something more with the drug. And so what is happening next is that, everybody thinks they can beat sunitinib. And so this study is also going to go into a second line study known as Strategist 3. This will be a phase 3 study. You'll either get IDRX-42, the new drug, or you'll get sunitinib. If you get assigned randomly to sunitinib, you can always cross over and get the new drug. So everybody who goes into the study has the opportunity for the new drug. It's just whether you get it first or second that is randomly decided. Importantly, for the future of the drug, IDRX company was acquired by GlaxoSmithKline. So that was a really large deal. So that will give additional financial resources for future drug development. Because unfortunately, sometimes drug development stops because companies run out of money. In fact, bezuclastinib used to be with a different company, and they kind of gave up. And a different company, Cogent, says we can work with this. So having financial resources is important, but right now we're trying to activate the study. But because of the integration, we're being kind of put on hold because GSK wants to know what's going on. Now, what would be the implication of this study being successful? Well, if IDRX-42 or whatever its name turns out to be better than sunitinib, then that would be the new second line therapy. But if the last study was positive peak, then you actually would now have 3 choices in the second line. Sunitinib, which is the original, sunitinib plus bezuclastinib, or IDRX-42. So again, it might get confusing in the future because we'll have to decide, sort of as an academic medical community and as a patient advocacy community, like which of these choices do we think we want to do? And if that wasn't complicated enough, there's another study that's trying to be the second line drug. So ripretinib, which is currently approved as the 4th drug, ran a study a number of years ago trying to beat sunitinib because it was felt that ripretinib was a better mousetrap, worked in the lab better, so that it should be able to beat sunitinib. And so this was a straight head-to-head comparison, and based on our phase 1 data, everyone thought ripretinib would be better, but it was not. So these are, again, another favorite data presentation mode of oncologists. So these are survival plots. And so in these plots at time 0, which is the start of the study, no one has progressed, right? they haven't even taken the drug. And so it's 100% progression free, or 100% of people haven't failed. But as patients have progression, the curves start dropping down. And so each time the curve deflects downward, that means that one person at that time point had progression noted and they went down. And so in these curves, you want to see them separate, and who's ever on top is the better treatment. And whoever's on the bottom is the worst treatment, as long as there's a difference that we believe is actually true. As you can see in these studies, the curves are right on top of each other. So there is no difference between these drugs. And because there was no difference, the FDA would not approve this for sort of, it gets complicated statistically. Proving that you're better than something is way easier than proving that you're not worse than something or that you're equivalent. That actually requires a lot more people. So most of these studies are like, we are going to be better, but if we're the same, no conclusion can be drawn. And so this study went forward a number of years ago, and if this had been positive, this would be the new second-line therapy, but it wasn't positive. But there was an extra part of the study that was conducted, and that is these shedding DNA molecules for the tumor, what we call circulating tumor DNA. We collected samples from everybody before they started treatment because we wondered whether or not, because we know there's these different mutations and we know these drugs work differently, were there groups of people who actually got benefit from one drug but not the other, or the other way around? And that turned out to be true. So if at your baseline you had a KIT exon 11 mutation and you had one of these AP mutations in 13 or 14 that we could detect in your blood, again, circulating tumor DNA, a quarter of the time, even when you have a lot of tumors, we can't find anything at all. But in the people that we could analyze, if you had the ATP binding pocket mutation, then actually see the curves are separated. Red is sunitinib. So sunitinib is a lot better than ripretinib if you have this actual profile. And in fact, your benefit was almost a year longer to get sunitinib. But there was a different population that had these AL mutations, 17, 18. So again, you can see the curves are even more separated. If you had this profile in your blood before you started therapy and you got ripretinib, again, there was almost a year benefit to getting ripretinib over sunitinib, and there was actually a survival advantage. And so the overall study was negative, but the company wondered whether or not they could salvage something out of this. And so there is a study called INSIGHT for people who have imatinib-resistant disease, so only imatinib previously, and we draw your blood, and you have this mutation profile of 11 plus 17 or 18, which is only about 1 in 6 people would actually have that profile. You get randomized 2 to 1. This study is very small, only 54 patients, because the effect is so strong, we don't need a lot of people to see the differences. But the problem is we need 54 people, but it's only 1 in 6 people that we screen will have this profile. So it's challenging because we are having to screen hundreds of patients to find an eligible patient. So the study's running. We don't really know with studies. You never know when they're fully have all the patients until they announce they have all the patients. Based on kind of historical projections and totally making this up, I would guess that sometime in 2026 we might have results. Again, if this is a positive study, this would be another second-line drug. And so now, if in the theoretical world, when all of these things worked, we would have 4 second-line options. Sunitinib, which probably nobody would take, at least in the US. Sunitinib plus bezuclastinib, IDRX42, or ripretinib, based on selected patients. In the GIST community, we're pretty interested in the INSIGHT study because a lot of patients and doctors order these circulating tumor DNA tests, and we really don't know how to use them. So this would be the first study that would say a test result defined a better pathway than what you would do otherwise. Because right now there's no data that, if I draw this blood test and decide to give you the fourth drug versus the second or third drug, that's smarter or better for you. And we would like to know if that's true, because we would like to be able to design other studies that utilize this technology. Okay, so there's a lot going on in second line and a lot to be sorted out over the next 5 years as multiple studies report and potentially multiple new agents are approved. The other big space right now for drug development is still in the third line and later. So there's another KIT inhibitor called NB003. Again, it doesn't have a name yet. Maybe it will. This is a waterfall plot. So again, looking at it as, just sort of as a layperson or just as an oncologist, even, you would like a lot more bars going down than going up, which is true. And you would like the bars to go down by a lot. Here's the line of 30%, and that's also true. So in patients who went into study, and you can't really read it, but like this patient here was on 7 previous lines of therapy, and there's lots of 4s and 5s and 6s. So these are people who have been through everything that's approved, plus some other things, and nonetheless, this new drug still has activity. But we don't really quite know where it's going yet, so it's no longer enrolling patients into the phase 1. They're likely, because this has been positive so far, they're likely to do something more because it seems like this drug could get approved. But are they going to go to the second line and be yet another study? Would they go third line? Would they go fourth line? Or would they go after fourth line, which then also kind of calls into problems of what's the study design and would it be placebo controlled or not, which we have a lot of issues with, but sometimes the FDA rules kind of enforce that type of thing. Another factor that leaves this a little bit unclear is that in these phase 1 studies, you do lots of doses and sometimes you go way higher than what you think you need. And so they haven't really said what the dose is. So we don't really know how to interpret all the safety data, which has been somewhat limited because it only gets presented a couple times a year. So we don't really know what the dose is going to be. If it's going to be second line, third line, or fourth line, it's going to have to be a phase 3 study. And then if it came in fifth line after ripretinib, then it depends on what they think is going to happen. If the response rate could be 20% or higher, you don't have to compare it to anybody. You could just go in, and if we get 22%, the FDA would say that's good enough. But if you think the response rate's less than 20%, they're going to make you do a phase 3 study, and then you're going to have to compare it to something. And that gets problematic because all of the studies have used placebo, and we don't like that, right? And nobody likes that. And historically, we've always felt like since people can cross over, that you still can get rescued. Like if I didn't get the second line drug, I can still get it, 3 months down the line and I'm still be okay. And that has generally been true until we got to ripretinib. And then when you get to the fourth line or later, it may not be true that we can let you progress on placebo and get sicker and then rescue you back. And so there has been an initiative in the academic community and with liferaft saying, we've done all these placebo studies, and every placebo study, the result is the same. Second line, third line, fourth line, fifth line. You can only go 4 or 5 weeks before you progress. So why can't we use all of that past data to make a sort of a made up placebo arm and say we have to beat 5 weeks by so much to be, statistically important. But the FDA has not embraced that type of thinking, and so we have some editorials with the liferaft group, and we're trying to change that, but we don't really know. And likely, sometime in the next few months, we'll get a press release from the company and they'll say what they're going to do next. And then they'll start emailing all of, MD Anderson and Knight and UCSD saying, hey, we have the study, do you want to participate? There is a new problem that's emerging, which is something called APAL, or Apple. And remember we talked about you could have AP or you can have AL, but now the new issue is that you have both at the same time. So historically we worried about AP situations or AL, but as you use more and more drugs, you force the tumor to work harder. Not that it's conscious or working, but random mutation will result in getting 3 mutations. So it has both the AP and the AL on the same protein, which is different than this 2 mutation versus this 2 mutation. This triple mutation is harder. And so what happens is in a KIT exon 11, you start at the beginning. So you have all the cells are these orangey KIT exon 11s, and we apply a drug like imatinib, and it has to find a way to escape. So it can get an AP mutation, or it has an AL mutation. So then you have sort of 2 sets of clones running in your body. You have the orange clones and the yellowish clones, and that's sort of the car key problem that some drugs work on some, but not the other. But then as you start applying more, potent drugs, then the way that this clone escapes is to get the triple, or the way that this clone escapes is to get the triple, and eventually you end up with all of these cells that are these Apple mutants. And we know that this is starting to happen. So if you look at people with drug resistance before ripretinib, and you biopsy them, a resistant tumor, and you go, well, what's going on in this tumor? About 1/3 of them will have AP mutations, and almost 2/3 of them will have activation loop mutations, but nobody hardly historically has an Apple. But if you take our 4th-line drug, ripretinib, so everybody who got ripretinib for the most part was on imatinib, sunitinib, regorafenib, maybe something else, and then ripretinib, and you biopsy the resistant tumors and ask what's going on, the AP mutations still persist because the drug's not super great against AP. It's really good against AL, so you get rid of that. But what you now see is that half of the resistant lesions are Apple. And that's a problem because we don't have any drugs that overcome Apple mutations. And so we've tried to look at all the drugs that have ever been used in GIST, or even ones that failed in their use against GIST, and none of them have worked against Apple mutations. So right now, this is an undruggable problem. How are we going to solve that? Well, one solution, which is kind of why everybody's rushing to the 2nd line, is get your best drug in earlier and try to wipe out the cells to the extent that you can before you get all these Apple mutations. But ultimately, even doing that, the Apple mutations are still going to arise. So we're going to have new drugs, and chemists are going to have to find new ways to treat these mutations. The other thing that we sort of want to figure out is, like, what is this happening? How can we identify somebody who has an Apple mutation? I won't go into all of it, but it's actually pretty complicated from a DNA sequencing standpoint, because when we do our DNA sequencing, we can do little chunks of DNA, and that's all we can do technically. And these things are really far apart, and so we can't prove that this are all together. We can prove that you have all of them, but we don't really know if they're an Apple or they're an AP mixed with an AL. So we're going to have to get better diagnostics to figure that out, but we haven't figured that out yet. So new drugs, so like pivoting away from kinase inhibitors, let's do something different. So there's a new drug, NN3201, which is an antibody drug conjugate. So antibodies are proteins that normally your immune system would make. So if you get vaccinated for shingles or COVID or whatever, you would make antibodies against what you got vaccinated. But it's possible to make antibodies against anything we want in a test tube. And so KIT, which is the protein that we've been drugging as an enzyme, and Dr. Lazar will tell you in a few minutes, is expressed by nearly every KIT, or every GIST. So all the cells have this KIT protein. And so people have figured out a way to take an antibody against something like KIT and then put a chemotherapy agent attached to it. And I'll explain how that works in a minute. So this is the clinicaltrials.gov number that you can look up the study if you're interested. You only have to have had imatinib. You don't have to have had all the drugs. It's opening this year, and maybe it's open at some sites. And the theory of this is that we don't really care about APL mutations or Indian mutations. All GIST, even ones without KIT mutations, could express KIT, 95%, and so we could target them. But we have to figure out a dose, and KIT, that's that door I showed you. It's not necessarily evil, like we talk about it like it's a bad thing, but your bone marrow loves KIT because a lot of your blood cells need KIT in order to grow successfully. So if we start targeting KIT with chemotherapy, are we going to kill your bone marrow? That's a thing. The other thing, and Dr. Benjamin was around doing chemotherapy studies, and he could tell you, and had many publications, that GIST do not respond to chemotherapy. So can this work? The theory of it is, and I'll show you, is that by delivering the drug directly to the cell and only releasing the chemotherapy in the cell, the theory is that we'll get a super high concentration of chemotherapy in the cancer cell, but not in the tumor cell, or not in your blood and the rest of your tissues. So the way I think about it is like the antibody is half of a Velcro patch, and your cell, the GIST cell, has the other half. So when we infuse this drug, it just goes flying around your body, and you will metabolize it. But if it runs into a GIST cell, Velcro sticks. And when it sticks, the cell says, hey, there's something stuck to me. What should I do with it? I'm going to eat it. So it takes it into the cell and puts it in something called an endosome. And then there's a change in pH that causes the chemotherapy to be released. And so the idea is that we get really super high concentrations of the chemotherapy in the cell, but it's not in the blood. But it's possible that if this wasn't designed correctly, this could leach off in your blood. Or there's also the situation, like if we kill cells super efficiently, then we release the chemotherapy back into your blood. And so these antibody drug conjugates, the dosing gets kind of very critical because we want to target the tumor, but we don't want any of the chemotherapy to leak back into your system. There's also other possibilities of how this drug could work. So there's the design intent, which is this antibody releasing the chemotherapy. But KIT is a receptor, so by putting an antibody on the outside of it, you may interfere with its function, and so we might get a little bit more activity from that. And it's also possible that by sticking an antibody on the outside of the tumor cell, the immune system, which had been ignoring the tumor, might say, hey, there's something with antibodies stuck to it. That's usually something bad. I should attack it. So we think that the chemotherapy attack is the main way that this will work, but it's possible that it will actually work via other mechanisms. There's another drug called ziftomenib, which is a menin inhibitor. So a very smart group of people at Dana-Farber, led by Matt Hemmings, did this whole huge project where they asked, if we take out one gene at a time or one protein at a time in a GIST cell, can we find things that will kill GIST cells but don't kill every other cell that we test? And the proof of this is if you take out KIT, GIST cells should die. So that part came true. But they identified that there was a dependency of GIST cells on something called menin, which binds to something called MLL. And in the GIST cell, this is DNA. These are histones, which are kind of proteins bound to DNA. And so this complex forms, so menin binds, MLL binds, and other proteins bind. And when all of that happens, the GIST cell turns on certain genes which make it very happy and make it grow very well. Ziftomenib, which has been designed actually originally to treat leukemia, because there's a whole group of leukemias that are dependent on MLL, but in GIST cells, this drug dissociates menin from the other binding proteins, especially the MLL. And so the thought is, and tested by them in a test tube at least, that if you treat the cells with this menin inhibitor, the cells will be unhappy. And this is a mouse experiment where there's a tumor in a mouse, it's a certain size here, and then you don't do anything, you give this like saline, so this is the placebo mouse. Or you can use this new drug shown in blue, or you can choose imatinib. So this is a KIT exon 11 mutant GIST. So imatinib has an effect. It's not a curative effect, but it really slows down the tumor growth. But if you do the combination, you get a lot more effect than either treatment alone. And so the theory is that even in a GIST that imatinib is not working perfectly against, that we are only partially inhibiting KIT because of these mutations, whatever we get out of imatinib, that should synergize with the ziftomenib. So there's a phase 1A/B study going on. The only inclusion criteria is that you have prior imatinib, and you can't have a specific mutation that makes imatinib not even bind, called KIT T670I. So those patients are excluded, but that's super rare in GIST. So almost anybody after imatinib would be eligible for the study. It's a little tough sometimes to figure out where it's open, but it's definitely opening this year. I expect it to open at our site in a couple months. I don't know. Same? Yeah. Yeah, so we actually have IRB approval, but we still have to do contracts, which is super slow. San Diego's doing it too. So, and if you look on clinicaltrials.gov, there is also a list of sites that are participating, so you can always, find the site closest to you. So this is a very interesting approach. Again, it's different than our typical study because it's not all about KIT inhibitors. And then, yet more, there's a new drug target in GIST called PRMT5. So PRMT5 is an enzyme, and it turns out by sort of a happy coincidence that most GISTs have deletion of something called CDKN2A. So CDKN2A is not the accelerator of the car, it's the brakes on the car. And so GISTs want to take the accelerator down and the brakes out. So they oftentimes delete CDKN2A. But stuck really close to it, like it's almost impossible to delete CDKN2A out without taking out something, there's something called MTAP. And MTAP is frequently deleted along, and that creates a metabolic vulnerability to the cell that's kind of unique to GIST. And you can target PRMT5 and turn off its activity. And we're not quite sure exactly all the things that PRMT5 does, because it does a lot of things, which is sort of good from a efficacy standpoint, because there's a lot of ways that the drug could work. Sometimes it's a bad thing, because if you target too many things, then it just becomes toxic. But it can change splicing of RNA, which affects the proteins it makes. It can also put these methyl groups on proteins that lead to degradation. And so one thought of why PRMT5 inhibitors could work is that they do all this stuff. But another theory is that we know that in many cancers, but not GIST, we can target the immune system. We can give immunotherapy, and in lung cancer or melanoma or renal cell cancer, we have these dramatic responses. In fact, there are people with advanced lung cancer or melanoma that historically would have said that their median survival is less than a year, and they are disease-free 5 years out and off therapy. And so we know that targeting the immune system can be very powerful, but just do not respond to that hardly at all. But there's some thought that PRMT5 will actually increase the immune response. So this is not a GIST model, this is a mouse model. Again, these are the survival curves. So basically when the line hits 0, that means all the mice getting that treatment are deceased. And so you have the control mice, you can get the PRMT5 inhibitor alone, you can get the immune treatment, which in this model does not work very well, not better than control. But if you combine them, all of a sudden you have mice that are surviving. So at least in this short duration of therapy, 80% of the mice are surviving because somehow the immune system has been alerted or woken up by the PRMT5 inhibitors. There are multiple drugs. This is again a phase 1 study. In this study, basically the only inclusion criteria is that you had the deletion on chromosome 9 of this MTAP. So there's one GIST patient that was treated, and their bar went down a little bit. I mean, like, I don't know what to say about it, but you know, there is reasonable activity for an early phase agent, especially considering that some people got very low doses. And that's kind of the upside downside of phase 1 studies, that in phase 1 study, we start at low doses. So if you come in early, you might be on a lower dose than what we ultimately decide is effective. But in other studies, the first or second dose actually is therapeutic, and so you want to be in. So it's kind of hard to know exactly what this means, other than it seems to be doing something. This is actually, it was interesting. There was only one GIST patient, but they highlighted it for immunohistochemistry. And in this column, brown is before treatment, and brown means that PRMT5 is not inhibited, but once you get the drug, it is inhibited, so all the brown cells went away. So again, this is very interesting. It has gone through multiple phase 1 agents that are basically cancer type agnostic. You have cancer and you don't have a treatment, you can be in the study. It does require specialized diagnostic testing because we have to confirm this deletion. Not sure, what the companies are going to do, but Canis Haddix, who's at Dana-Farber, is planning a study with a KIT inhibitor plus a PRMT5 inhibitor. And it's still kind of being worked out, the design, and we still have to get the company to agree to, fund it or at least give us the drug. So don't know what exactly to say here other than the next big sarcoma oncology meeting, CTOS, Connective Tissue Oncology Society, will be in November. And so likely there'll be some chatter about that, and that will maybe spill over into GSI and liferaft group, newsletters or posts. And finally, I wanted to talk, and Nita showed this slide before. So this is BFR14. So these are patients with metastatic GIST treated with imatinib, and all those people who went 3 years without progression. So ignore everybody whose results weren't as good. At 3 years, they were stopped randomly or not. And she showed the curves, and basically if you continued therapy, that was a better outcome than if you didn't. Most people who stopped, in fact, 84% of people who stopped by 2 years, they had progression and had to restart the drug. But what's happening in these curves is not the same thing at all. The top curve is what I've been talking about for most of the last hour, which is resistance. So you keep going on a drug, and over time, the drug doesn't work because the tumor's mutated. So that's secondary resistance. But this curve is something different. This is persistence. This is hibernation. These are cells that were treated for 3 years, and if we took out their tumors, a lot of the cells would be dead, but we'd still find live cells in there. And the problem here is that we can never get to a cure if we can't stop therapy. And we can't stop therapy if cells can hibernate. This is 3 years, but there are similar things for 5 and 10 years, where 10 years of greatness on imatinib and you stop, and you still have the tumor come back in most people. There is a little bit of a tail, so maybe 20% of the people treated for 10 years actually are cured. We don't know, but that's the problem. And so it's quite different because it's not a resistance mutation. We don't understand this. We don't have a way to measure it. Resistance mutations, we biopsy something, we sequence it, we know what we have. Persistence, we don't understand that. We can, however, model that. So this is just a little experiment for one of my graduate students. So this is a GIST cell line grown in culture, and the y-axis is like how many cells were in the flask. Like we count them. We count a little bit of them, and then we multiply by the fraction that we counted and figure out how many were in there. So if you put imatinib into the culture, so like entering therapy, you can see that the cells go down a lot. We kill off 95% of them, but we never get rid of the last 5%, no matter what we do. And if we stop therapy, within a week, the cells have all grown back. Now, if we continue therapy, which is what BFR14 showed us, like we can maintain a small number of cells that we never get rid of. And we've done this experiment. It's kind of hard to do because eventually you get contamination, but we've done it for 6 months, like drug the cells for 6 months, and you take away imatinib and the same thing happens. So one theory about why this happens is something called autophagy, like self-eating. And so cells have a survival program called autophagy where they sort of cannibalize themselves and put themselves into a hibernation mode. There's some protein enzymes, ULK1 and ULK2, which regulate this. DCC3116, again, numbers, not names, is a novel inhibitor that inhibits these. And in mice, if you do the combination, you get more mice who appear to be cured within the limits of a mouse model, unlike if you just did a ripretinib alone. So this study is actually being run right now. It's a phase 1/2 study. We're combining ripretinib, which is the fourth line approved agent, with DCC3116, and we're still trying to figure out the dose. We know the ripretinib dose. We just don't know what the DCC3116 dose. It's open at multiple sites. You can look it up in clinicaltrials.gov and figure out where it is. We're running it. I don't know. Is it at MD Anderson? No. But I think it's at Miami. It's a lot of different places. And so once the dose has been determined, then it's going to go into an expansion where, like, okay, now we know the dose. Let's treat a bunch of people in second line. Again, this focus on the second line, and to see how that works. But we don't have any results. So to summarize, I had a lot to say because there's a lot going on. We're making a lot of progress. We have, better, stronger, more potent drugs that overcur mutations, and we're moving them into second line. There's been a lot of discussion, like, could we go frontline? And, ideally, some of these new drugs compared to imatinib probably would work better than imatinib. The problem is that imatinib is generic, and you can buy it from Cost Plus Pharmacy, Mark Cuban, for $35 a month. So the business incentive for someone to develop a frontline drug is very limited, because how much better does it have to be that you can charge way more than $35 a month? That's challenging, and that's just the US, where if we approve things, then Medicare and insurance has to figure it out. But other countries do approvals differently. They approve that the drug is safe and effective. So you told us what it does, we believe you. Our government will not buy it. So if you can buy it yourself, it's allowed in our country, but we're not paying for it. So even ripretinib is not yet approved, like in Germany or the UK or other countries. And so going frontline is complicated. I don't know if we're ever going to get there. Another significant point is that we're targeting new pathways besides KIT, and so we've made a lot of progress targeting KIT. It's been a quarter century. It's probably time for us to do something different. And it also logically holds that if we have better drugs, why do we make you go through drugs that work less well and have more side effects to get to them? Let's move the new drugs earlier, and that's what's happening. So this is my manifest. Thank you.